Hi, everyone. Good evening and welcome to Shoals Marine Lab's Rock Talk, or otherwise known as our Marine Seminar Series. Greetings from a very stormy Appledore Island, as all of those who are out here know tonight. I'm Dr. Jennifer Seavey, the Executive Director at Shoals. For those of you who are joining us from off island who are not maybe so familiar, we are the country's oldest and largest undergraduate focused marine lab, and we are jointly operated by Cornell and UNH. We're located in the Isles of Shoals on Appledore Island, about 10 miles offshore of Portsmouth. Every summer, especially this one, we are excited to have our students on the island, and we offer our island community these weekly Rock Talks, and we're really happy to have all of you here with us tonight via this Zoom format. So tonight our talk will be 45 minutes, followed by 15 minutes questions and answers. If you want to ask a question, please use that Q&A box that's at the bottom of the screen. Type in your questions, and then I'm going to read them out loud to the speaker at the end. So I feel like many of you actually know this speaker tonight, Dr. Chris Kenaley. He is an assistant professor of practice in the biology department at Boston College. He has his undergraduate degree in natural resources from Cornell and a PhD in aquatic and fisheries science from the University of Washington. As a comparative biologist, Chris studies the evolution of feeding and swimming systems in fishes. Specifically, his research probes two questions. One, how form and materials dictate performance in the biomechanical systems of non-tetrapod fish, and how the acquisition of key biomechanical properties has influenced the evolutionary success of this group. He kind of wins the prize for a really great talk title. Skin, Scales, Fangs, and Waveforms, Multidisciplinary Approaches to Studying Fish Biomechanics. So without further ado, I'm going to invite Chris onto the screen and he can share his talk with us. Thanks, Chris. Welcome.
Thank you, Jennifer. That was such a nice introduction. It's really a pleasure to be here at Shoals, at least virtually. I grew up in the area. I was telling Jennifer and Colin this before, so this is a real honor. It's also great, even though it's one way right now, to present some work in front of some familiar colleagues. So with that, I'll share my screen. And okay, so hopefully we'll get through what I want to here in about 45 minutes, and then I can take some questions. The way the talk is arranged, it's really a series of vignettes, 4 projects, a description of 4 projects that my lab has been involved with over the last few years. I run an undergraduate lab. That's my job at Boston College. So everything you see here has been led by undergraduate students at Boston College. And just to set the scene here for how students usually get going in the lab with me, well, we have 9,000 undergraduates at Boston College. A lot of folks don't realize just how large Boston College is in terms of its enrollment. 1 out of 10 students is actually a biology major, and almost 3/4, or at least 2/3 of them, are pre-medical concentrators, folks who have aspirations to go to medical school. And we require an advanced experience, an AE requirement for our undergraduates. That is some sort of research experience before they graduate. Okay, it's part of the degree requirement in the biology department. And so we have about 200 students that roll through faculty labs, including mine. I have about 8 right now, 7, 8 at last count. And so given the demands of applying to medical school and also our AE requirement, I hear this a lot from my advisees, folks who've taken my classes. I need some research experience, right? And if I have space in my lab, I love working with undergraduate students, so I usually take a few on each year. And the goal of the research in our lab is to study biomechanical systems, right? And I think this is really great, even for folks who are not going to study biomechanical systems. It's by definition, uh, I guess my talk title is a little redundant. Biomechanics is a multidisciplinary field uh, by its own right. Right? You need some understanding of engineering, and nowadays computer science, and then if you're going to analyze the data, some data science. But it ticks a lot of boxes for undergraduates with some skills that they can take with them into their future careers, even as folks who are practicing medicine. And I just want to reiterate that this field of biomechanics, some in the virtual audience here know this. It's a very multidisciplinary field. And all you have to do is open up the standard textbook by the late and great Steve Vogel, and you'll see applications of models, often very simple ones. Also, he describes in his text how he performs his own experiments to demonstrate different principles in biomechanics and how he just made things. And so through the talk, you'll see just how the undergraduates in my lab have done just that, apply models or made things so that they can answer questions. And that's really what biomechanics is all about to me. Finding an interesting question and then making something or applying a model to answer that question. So the first, as I mentioned, be broken up into 4 little vignettes here, 4 little descriptions of some projects that we've worked on recently. The first one will be the role of skin in locomotion, all right, in fishes and rayfin fishes. It's an important tissue. It covers all the musculoskeletal linkages in the body, right? And if these linkages are to function, it must deform, all right? But there's an important catch there, in that it's full of collagen. And collagen's a very stiff material. So one function of fish skin could be that it transmits force, right? Having all this collagen and being rather stiff. And here's a figure from a paper we published in the lab, or out of the lab a few years ago, where we just looked at some real simple anatomy of the myotomal structure in a few fishes. And what I'm showing you here is how these myotomes, the muscles of the body, of the swimming body, are connected to the skin by some tendons. And that skin is full of these collagen fibers, as I mentioned. So with this project, our goal was to, figure out, is that skin transmitting force, right? Is that skin part of the swimming system, or is it just there to deform freely around the body and really not get in the way? So 2 undergraduate students who joined the lab were really interested in this. When I described this question to them, they really got into it right away. And Andres, he was the first to come on board. And he said, it sounds to me like skin shouldn't get in the way. Swimming fish need stretchy skin to accommodate the bending, right? And so I just showed you this video of a trout swimming. You'll see more trout swimming later. The body bends and the skin expands about 10%, right? And so you can make the case that skin should just be kind of flabby and not very stiff, not get in the way, right? But then Janelle came along and said, but what about the structure in that myotomal system connecting to the skin and all the collagen fibers and so forth? Maybe it's there to transmit muscular power, transmit the force of the muscles along the body. And so that's what this project was all about, trying to answer that question, its functional role in swimming. Is it there just to not be so stiff and not get in the way, or is it there to transmit force? And so we looked at a few species of fish. We started hacking them up right away. We looked at red snapper, coho salmon, Florida pompano, and these things, they don't represent any special diversity. It's just what we had kicking around the lab and fish markets, things we could catch. On vacation, say I was down in Florida and we caught some red snapper and some pompano. So what the hell, it just brought them back to the lab. And for the red snapper, we cut them up 5 different pieces and stretched the skin. But Andres, he said, yeah, these are kind of big. Let's do, at least for the coho and the Florida, he said, well, we can look at skin samples at a much higher resolution. So we had this great idea of putting this coho skin on a piece of cardboard and cutting them up in little pieces. You can see them here, and then marking their position, right? And we digitized this piece of cardboard afterwards, and you'll see how that was important. And we did the same thing with the pompano. So we got, you know, from 2 coho salmon and a few pompano, we have somewhere close to 150 in the coho and around 40 samples or so in that in the pompano. And so to establish how stiff those skin samples were from across the body, we built a machine. We built a testing rig, all right, a material testing unit. Very simple. It runs on a $20 microcontroller, an Arduino unit. That's attached to a force transducer and a linear actuator. That is a simple motor. And so we just stretched the skin and measured how much force it took to stretch the skin a certain distance. That is, looking at the stress and strain relationship of the skin. And you can see here the results of some of these experiments, or at least one result from an experiment. But this is what it looked like as we were stretching. And it just pulls on the skin and it measures the force. It's pretty simple machine, very simple procedure. Okay, and so we get data that looks like this. We estimate the strain, how much the skin is stretched. And then also we're looking at the stress, how much force for cross-sectional area has been applied to the material. And then from that, we establish stiffness. Now, biological materials are interesting in that they don't often have these linear stress-strain curves. And so we looked at stiffness at 3 regions across the strain of the material. First at 10%, then at 20%, and at 30%. And so we have 3 stiffnesses given a certain strain value, and you'll see those come up in the next few slides. So stiffness, just to reiterate this, the stiffness is just the relationship of this stress-strain curve, or it is the relationship between stress and strain, stress divided by strain. And so these, just getting right into the results, and I'll do this for the next, for the rest of these little vignettes as well, just getting right into those results with a little bit of background. So I won't show you any data from some of the snapper material, but from the pompano and from the coho, we saw some really interesting results, in that the skin was certainly stiffer in the posterior, and this is just a heat map of that stiffness at those strain values, 5, 10, and 15% strain. All right, and then what I'm showing you on the right here is just a linear regression of those of the stiffness and the position along the trunk, the percent of the trunk. So down here at 0 would be the head, 100% would be near the tail. Okay, and so what we see at 5, 10, 15% strain, at all of those strain values, we see stiffness increase along the length of the body. That is, the skin is stiffer closer to the trunk. Okay. And from that, we inferred that, well, stiffer skin in the posterior of the trunk indicates a force transmission function, all right? That is where thrust is produced, and I'll say more about that in another section of the talk. That is where thrust is produced, right? And that's where force needs to be transmitted from the myotomes of the trunk for thrust to in fact be produced. And so Andres Sannon and Janelle Ackerman led this and published this a paper in the Journal of Morphology that reported these results, that skin is a really effective force transmission tissue, right? It's not just there to accommodate bending, it actually forms a really important functional role. So the next thing that I'll talk about is a project led by another student in the lab. I'll mention her name in just a second. And that's scales and their role in defense, all right? And this is another materials project. And scales are a really hot topic, especially for engineers. There are many engineers thinking about how scales can inspire new human armor designs. It seems like every time I click up Google Scholar, there's a new paper devoted to scales and fish scales and their material properties, how it has inspired some design system engineers looking at this material all the time. And there was an important paper published last year in this new journal Matter, where These folks proposed that these massive, very thick scales of this rather endangered fish in the Amazon, the Arapaima, as a necessity for the protection of that fish, protection from piranha attacks. In other words, they live around a bunch of piranha with these nasty teeth, and these scales are defensive mechanisms, defensive tissues to prevent damage from these piranha. All right. And this was interesting because when this paper was published after the student and I started working on this project, and as far as we could tell, no one's really assessed the defensive role of scales. Are they really armor, right? And I'll say more about that in a little bit, but this caught our interest. And then a month or two later, some other folks published a response to this paper saying, "Whoa, this is crazy. This is crazy. There's no way that these scales evolved to, in some way, be a defense against piranha." They cited some phylogenetic evidence, some biomechanical evidence, feeding behavior, more or less saying that piranhas arrived on the scene well after Arapaima did. That piranhas don't have mouths big enough to wrap around an Arapaima. And matter of fact, there's from gut content analysis, there's no evidence that they do eat Arapaima at all. So this didn't necessarily call into question that that scales are defensive tissues or armor tissues, but it's just it kept us going with our research, understanding that there's still a lot to learn about scales and their functional roles. So this is Eva, the student who led this project, and she, when she got going with it, she asked this or stated the obvious here: if scales are indeed armor, they'd be more puncture resistant, where that property is most important. That is, if scales are indeed armor, they would provide a defensive function around the areas of the body where that function would be needed most. Okay. And no one had tested or investigated scales in this way before. And so she focused on two areas of the body that are really sensitive and really important to a fish's biology. The first being the lateral line, where flow is sensed on the body. And then also where the gut is, okay? And what we did is we broke up the fish into, all the fishes that we studied into 4 quadrants here: upper left, upper right, lower left, and lower right. And it's the lower part of the body, as we all know, especially those taking Nick's course, this is where the guts are on the fish, all right? And so our hypothesis here is that the closer you were to the lateral line, or the closer the scale was to the lateral line, or if the scales were over the lower part of the body, they would indeed be more puncture resistance. And that would be an indication that scales are indeed used as an armor defense. And so to get going with this work, Eva, who is a double major in journalism and biology and had no experience in any sort of mechanical engineering before, I asked her, well, you need to build a testing rig. If you want to do puncture resistant tests on scales, then you got to build a testing rig. You know, we have to push something through these scales that mimic a tooth or something like that, right? And she said, sure, why not? And so over the course of a week, we built a testing rig. It looks like this. Here's a schematic of it. I'm going to move the screen of your photos here, just describe what's going on here with this rig. We have a very simple motor, a linear stepper motor, right, that pushes on a force transducer that's connected to a needle, all right? And so this just plunges a needle through this tissue, all right? And here's a close-up of a needle. It was a fasting needle, something you'd use to fast 2 pieces of fabric together. All right, and we changed these out for every experiment. We went through thousands and thousands of needles. And so she pushed these needles through the scales that were on the fillets of the fish that we had studied in this project. And she was very clever about keeping track of where these scales came from on the fish. She just made a grid. She took a picture of the fish, of the fillet that she was testing, and made a grid. And so you see, it's hard to make out here, but the little pieces of text here indicate the centroid on about a ½ centimeter, or if the fish was large enough, maybe a centimeter square piece of the fillet, right? And so she just put the fish underneath the needle where she wanted to test and recorded the data. And then she was able to come back and say something about where that test was from and how puncture resistant the scale was from that location on the body. So this is what the data looked like from the some 2,300 experiments that she did, 2,300 puncture experiments. Maybe you do know, maybe you don't know that the scale system in fishes is layered. And you can see this from the results of this one experiment here, where the needle plunges through the first scale, right? The force increases and then the scale fails, it starts going through the second scale, and then it fails again. And then it goes through a third scale. So in this case, there were 3 scales. There could be 4, there could be 2, and rarely there would be 1 scale. It's an overlapping armor system. And so from the force trace, we took the maximum force, all right? So we wanted to know on that area of the body from where this puncture resistance test took, what was the maximum force it took to plunge this needle through the body. And we worked on 6 species, all right? I won't show you any pictures of the species. They're all things that you could catch here around Boston in freshwater or saltwater. We had some centrarchids in there, sunfish, a few species of sunfish, largemouth bass. We also included striped bass, yellow perch, rock bass, all very common things around here. So 6 species in total. So I'll just jump right into the results. And what she found in general is that indeed the closer you are, even over these sensitive areas of the body, what we're postulating is sensitive areas of the body. I don't think it's a stretch to say that they are. But say, if you look at the distance from the lateral line, for all 6 species, right? I'm showing you data for this is the model output. These are the mean puncture resistance, the F max values that I showed you before, for all these species, according to how far that puncture resistance test was from the lateral line. So this is the closest lateral line position to that puncture resistance test. And you see in 4 out of the 6 species, we see a pretty strong relationship here that the puncture resistance decreases as we move away from the lateral line. 2 of those, it's kind of equivocal in a rock bass here, and then in the yellow perch, it seems the opposite case here. But in 4 of these 6 species, we found evidence that the puncture resistance of scales close to the lateral line, and we certainly included some lateral line scales in that testing as well. Those scales were much more puncture resistant. And then as far as the quadrant, just to recap here, we expected the lower half of the body to be more puncture resistance. And indeed, we see something of a pattern there as well, that for the most part, the lower parts of the body where the gut are, the more ventral parts of the body where the guts are more puncture resistance in all 3 species. So you're seeing again on the y-axis, these max force values according to the quadrant, lower left, lower right, upper left, and upper right. And I've just highlighted these to the lower parts, the lower quadrants, the more ventral quadrants, to show that indeed for most of the species, those are the ones that have the higher puncture resistance values. So in general, it looks like scales are more puncture resistant near or over sensitive systems, all right? And it indicates a protective role. And some of you might be saying, yeah, well, we kind of know scales are armor, they should be protective. But no one actually really tested this before. And that was the beauty of Eva's first question, like, well, let's actually test this. And so I think she's the first to really, in a very exhaustive way, demonstrate the scales are actually used as armor in fishes, right? Even the very small ones on these species had no, they don't have scales like that Arapaima. They're just, your standard fish scales. Okay, so the next little vignette here was led by Ernie Violet. And Ernie came to me after taking my physiology course. It's a comparative physiology course, and I spend one afternoon in that course, one lecture talking about deep sea fishes. It's my dissertation focused on the evolution and biomechanics of feeding in a few groups of deep sea fishes. And I'd like to share just as much as I can about the deep sea realm, because these are pre-med students by and large, and they more or less haven't taken the time to think about it, or haven't been exposed to any sort of science about deep sea fishes. And so in this lecture, I share that when I was a graduate student, even before I was a graduate student working at Harvard's Museum of Comparative Zoology, that I had a chance to go right off in New England here and catch a bunch of deep sea fishes. Matter of fact, this Bear Seamount, where we've done most of the work, some of that work that we did do, it led to the creation of the Bear Seamount as a national marine monument. Now it's protected. And this is a 3D image of what Bear Seamount looks like. It's an extinct volcano from 100 million years ago, but it's pretty close. It's right off here, and it's extremely diverse in terms of deep sea fishes. And this really got Ernie excited. I showed him this picture, as I did everybody else in the class. And right away after the class, he said, "Oh man, that is just the coolest thing I've ever seen. Look at all those big teeth on those fish." And these are, we're going to be talking about dragonfishes. There's only one dragonfish, and that's right here. But you see, in most of these other things, there's just these massive fangs, right? We have a few anglerfishes here. Here's an anglerfish of the genus Linophryne. Here's a Knocklegaster. It's a pretty iconic deep sea species. But you see, most of these things have these massive teeth. And Ernie, he asked a question that I was thinking for a very long time. Like, what are those teeth doing? Are they slamming these teeth through their prey items, or just how do they use these crazy teeth? And Ernie said, I remember looking at your website, and that you had worked on the teeth, or at least the jaw system of deep sea fishes, and he was really excited about this species here. This is colliotis. Many of you know this species, I'm sure. It's again a very iconic deep sea species. This is Sloane's viperfish, and you can see the massive fangs on the interior of the lower jaw there. There are also a few in the upper jaw. So this is a micro CT reconstruction of the head of one of these animals, of Colliotis sloanei. And in some previous work, what I showed was that the biomechanical system here that closes the jaw, specifically the muscles, are really puny. All right. So this looks like a big muscle here. This is one of the big adductor muscles on the side of the head that inserts on the Mechelian fossa, the lower jaw. And that's the muscle that does most of the work in closing the jaw. It looks big, but these muscles are only just a few fascicles thick. Matter of fact, they, as far as if you know anything about physiological cross-sectional area or anything like that, you've ever done any work in that realm, these things have essentially no physiological cross-sectional area to offer in closing the jaw. It's very puny muscle. In addition to that, the mechanical advantage of this jaw system is super low. It's like 0.12. That's a really low mechanical advantage. That means any bit of force that is inputted on the jaw, in the best case, only 12% of it is going to come out at the end of the jaw. All right. So Ernie picked up on this in this paper that I wrote years ago, and he says, "Well, God, how are they using these teeth?" And aren't these jaws just too weak to pass a fang through anything like their prey item? And I said, "Yeah, wow, someone actually reads my papers, undergraduates no less." Colin. I just froze for a second. I think I'm okay. I'm okay. All right. So here's some of the results from that paper that I've been talking about for a while now. This is the bite force. We're modeling the bite force using a very simple mechanical model of the lower jaw system. And these are really low values of bite force. And so Ernie started his project from this point of view. Okay, this jaw system is weakly powered. Given that, what are these teeth doing? Could they be indeed puncture devices? Are they so sharp that they go through the prey, even though the jaw is weakly powered? And if not, what are they doing? And so just to give you a sense of of their prey, it's an interesting problem taken on their prey. First of all, they're myctophids. The myctophids, they're kind of like the goldfish crackers of the sea. Everybody eats them. They run the pelagic ecosystem in terms of prey for the piscivorous things that live in the ocean, whales, dragonfishes especially. Matter of fact, dragonfishes, for all the ecologists in the crowd, there are some estimates that they might eat twice the standing stock of myctophids in low latitude systems. So this would be like the Sargasso or the Gulf of Mexico. So dragonfishes eat a lot of myctophids, right? And this is what their prey looks like. You can see it's covered. This is Diaphis. It's a pretty common member of the midwater community. You can see these big scales here. Some of them have been lost. They're easily deciduous. They get lost easily, but you can see some big scales here, all right? So they eat a lot of myctophids and they're scaly, all right? The other thing is that they eat big myctophids, really big myctophids in proportion to their body, all right? So the mean prey size of dragonfishes, this group of some 400 species or so that all live in the midwater, the mean prey size is about 40% of the body length. That's pretty big, all right? And then the max is about 70%. And for colliotis sloni, that species I just showed you, the prey size is, the max is about 50, there's one report of about 60%, all right? So we're talking, big prey items. This isn't colliotis sloni, this is another genus of dragonfish. But here's the tail and here's the head of a myctophid that is in the stomach. Right? You can just see how big that prey item is compared to the body. All right. So you look at something like this and you say, there's no way they're impaling their prey with these teeth. First of all, how do they get them off? But then, impaling may not be the best device for getting an animal that's so large. You'd be attached to a big thing wiggling around forever. But they eat big myctophids. That'll be important, as you'll see in just a second at that point. So Ernie saw what Eva was doing, and he said, you know what I should do? How about I just test this in some very straightforward way? What if I took the teeth of some dragonfishes, of colliotis, snip them off from some preserved specimens, embed them in epoxy, and then attach them to Eva's testing rig, and then get some myctophid scales from the genus Diaphis, and see how puncture resistant they are, right? How much force, in other words, does it take to get a colliotis tooth through a myctophid scale? And what I'm showing you here on the right, on the x-axis is the size of the myctophid from which the scale came. And on the y-axis, I'm showing you the force it took. This is a lot of force. This is much more force actually than Eva's scales, all right? So myctophid scales are really durable and really puncture resistant, all right? And so the question is, does colliotis produce enough force to put its tooth through the side of a myctophid, all right? From these values, right? The answer is no. And so to answer that question more fully, Ernie took all this information together. The puncture resistance of the myctophid scales, the amount of force that we expect colliotis could produce given the physiological cross-sectional area of its adductor system, its mechanical advantage, and a few other parameters. And then the size, right? If I go back here, size does matter. If you're a bigger myctophid, you have more puncture resistant scales, all right? So we took all that information into account and built a model, right? And what I'm showing you here on the right is it's a complicated figure, but when Ernie made it and he explained it to me, I was really excited because it's just beautiful. It shows exactly what is going on here. So on the x-axis, what I'm showing you is the standard length of a colliotis specimen that we ran through the model. And from that, we predicted its jaw output force, what it could produce. All right, and then on the y-axis, what's being presented here is the prey proportion, the proportion of standard length. So a 50% prey item, let's say, on down to 20%, up to 60%. And then what's colored in here, these isobars, it's the amount of force, excuse me, yes, it's the amount of force it would take to puncture a scale from an animal that was this prey proportion of a myctophid that was that myctophid, that was that size. And then these other bars here, this is the amount of force times 1,000 that colliotis would produce at that given size, right? So the upshot here is that, say, a 20-centimeter dragonfish, colliotis, if it's eating its max prey size of 50% the standard length, it would have to produce, all right, it's right on this line, 4,000 times the amount of force it actually does with its jaw, all right, as predicted by our models, all right? So a 20-centimeter dragonfish is not putting its teeth through a typical prey item. So the story doesn't end there for Ernie. He came back to this after taking a break for a semester or so, and said, okay, well, what are those teeth doing? And so he took this jaw closing model that we had developed and added prey to it, right, as a mass and as a shape that would include drag being moved through the water if the jaw were to close on it. And in the simulation to the left, what I'm going to show you is the angular velocity and then the drag, the total drag that the jaw system is experiencing, right? So I'll just play that simulation. And what you see is what we did here is we just said, all right, what if the prey was impaled? All right, the two teeth went through it here. And it was positioned against the lower jaw, all right, flat against the lower jaw. Pretty much how we figured it would be arranged if it was going to be impaled, right? It's probably not going to be impaled on the thin edge. So I'm showing you that cross-sectional shape as it moves with the jaw. What you see is the angular velocity just plummets after there's an impalement. In the second simulation, Ernie flipped the prey item over and let it sit between the teeth, the two big teeth in colliotis, all right? And sure, the angular velocity did drop when there was a collision with the jaw, all right? And there was quite a bit of drag, but the jaw closing time was almost half of what it was if we assumed an impaling, all right? And so from this, Ernie put the pieces together, right? The fangs are powered by jaws too weak to puncture scales of the typical prey item. But if positioned correctly and unimpaled by some additional model, he figured out that the jaw performance increases substantially. That is, these might be caging devices, not impaling devices, right? That is, they might hold the specimen while the jaw is closing, not impale it, the prey specimen. Okay, so the last little vignette here is to share some work that Kylie Kramer and Haley Kunkel finished up about a year ago. And this is a little far afield from some of the subjects I've just talked about. There's no materials here, there are no fangs, no scales, and no skin. What they were interested in, they asked this question, they took a biomechanics course with me. And we study, of course, fish swimming in there. They said, how come nobody talks about propulsive wavelength? Everybody's talking about amplitude and frequency in swimming fishes, right? So they've read all, not all, but a few of the classic papers. And so we decided to study wavelength, propulsive wavelength, just kind of in some exploratory way, right? So what's going on with wavelength? Does it change with speed? Does it change with body position? And so we did all our work with rainbow trout, and what I'm showing you here is what I mean by wavelength. And so if you swim the fish steadily and you find the midline of the fish and you compare that midline to the axis of progression of the waveform, this is the waveform part of the top, this propulsive wave is going to move through the body as you see here, right? And relative to that axis of progression, that waveform has a wavelength, all right? And our question was very simple at first. How does that change with body position, all right? And some other things like speed, but I'm gonna focus on body position. So this is important in understanding how fishes produce thrust when they swim. You might say, all right, fishes wag their tail, that's how they produce thrust. It's a little bit more complicated than that, and there's some other things going on there. But in general, you might have heard folks refer to this as elongate body theory. There's a long body and the tail is whipping back and forth, right? It's undulating as the propulsive wave moves through. And we'll come back to this at the end of this this part of the talk. That wave that goes through the body, what it's doing is it's positioning the tail to push against some bolus of the water, some area or some volume of the water. And as it pushes on that and accelerates that water, there's a reaction. We think of this as some Newtonian thing. The fish is pushed forward. That is, some thrust is developed. And when folks have thought about this elongate swimming, they think about things like amplitude and how often that amplitude comes along, the frequency. That will predict how much thrust is being produced. But there's nothing about wavelength. So our goal here was to do some exploration of what's going on, but see if this could be in some way important. That is, wavelength could be important in understanding how fish do indeed produce thrust. And so there hasn't, as I mentioned, there hasn't been much done in this realm, how propulsive wavelength is implicated in producing thrust. There have been a whole bunch of studies that look at amplitude and frequency of the trailing edge, the caudal fin. But it took a lot of time to find those that, in addition to reporting amplitude and frequency, they reported propulsive wavelength, right? So there just hasn't been much work done. So Kylie and Haley had they had something from the very beginning. It was going to be new because just folks hadn't worked on propulsive wavelength, at least without any rigor. So when they started the work, they said, Chris, you're obsessed with R. Can we, and I am, I have to admit it. I'm not closeted about it. The whole lab runs on R. Can we do this in R? Can we say video record some fish swimming, rainbow trout swimming, and get the wavelength from that, from those video data? And I said, yeah, sure. So we spent the summer just writing an R package to do this. And so this R package very easily for your standard resolutions could get 400 points along the midline and also retrieve some high resolution contour data. So the way this works is we have our swimming trout and the package changes that video to some binary form. And the white area here is the region of interest, that is the fish. And we take that contour and we establish the midline, what we need for the waveform, right? And with the waveform, we can establish the propulsive wavelength. So we, as I mentioned, we're swimming rainbow trout. We're studying rainbow trout. We swam 5 juvenile rainbow trout and swam them at a broad range of speeds, about 1.5 to 4.5 body lengths a second. And with this new R package, Kylie and Haley were able to retrieve 240,000 body midlines, right? Not a few dozen or a few hundred, 240,000 midlines from thousands and thousands and thousands of frames of data. And this is just to get right to the results. This is what it looked like. I'm showing you here the smooth prediction from a GAM model, a generalized additive model, of the propulsive wavelength as it varies with body length, all right? It increases down the body, and here's the raw data here over 6 speeds. So in all our trout, the propulsive wavelength increases as it moves down the body, all right? And so the next question was, why does it increase? Or why, you know, is that important in swimming? And to focus, or to answer that question, what we did is we took that contour data and used those data to estimate what was happening on the surface where in other words, the muscle of the fish, right? So looking at the surface strain of the swimming trout, okay? And so I'll focus on just a few of these pieces from the figure. What I'm showing you here is the contour of the trout that is swimming in 2 points in time separated by a frame or two. And the propulsive wave, this black line, is moving down the body, right? And with that contour data, we could estimate the muscle strain or the surface strain, a proxy for a muscle strain. And the important things here to understand is that this wave, as it moves down the body, all right, it starts kind of a small value, about a third of the body length, and then it increases to 40% the body length when it gets about 2/3 of the way down the body. So what's happening there? What's causing that? Well, this muscle strain pattern that I'm showing you kind of explains that. I'm going to move you all out of the way so I can see this legend. So the red on the surface here indicates shortening, and the blue indicates lengthening. All right, so this is the strain rate. Okay, and so as the wave moves down the body, all right, that wave continues to shorten and shorten and shorten. Right. In other words, the behind the peak of the wave here and beyond the peak of the wave, there's just a whole bunch of shortening. Right, the whole wave is being shortened, right, or at least the muscles surrounding that wave are being shortened. And so in the next slide, I'll explain why that's important. But just keep in mind that this wave is sort of being changed. Its shape is being changed by the muscles on the exterior of the body. Okay. So this is how these two students would explain it to me, and how they did in our lab meeting when they produced these figures. Think of the wave moving down the body, and as it moves, the posterior of the wave is shortening, and it's pulling on that wave. This is muscle pulling on the body. All right, the muscle surrounding that backbone, if you will, or that midline is in the anterior. Excuse me, the anterior of the wave, closer to the head. All of that tissue around it is shortening, right, on the right side if you're looking down. And there's not much lengthening beyond the peak of the wave. And so what it's doing is just sort of pulling this wave up and out, right? And because of that, the wave is just lengthening along the axis of progression, right? And this was the cool finding, or what I found so cool about this project, is that is muscle power working on a body wave, pushing against, if you remember, that bolus of water, and then that reaction force is going to push back on the fish and provide it with some thrust. So that the increasing wavelength is indicating where thrust is produced. It's getting longer to produce more thrust at the trailing edge. So I'm coming up on time, so I'd just like to wrap it up and just, well, really sum it up. These projects are just, they've been a ton of fun for a number of reasons. One, these are just all exceptional people. It's just been a lot of great work. But in all of these cases, the folks have built something, applied a model, something they probably didn't expect to do is pre-med concentrators when they joined the biology program at BC. So, you know, and just to say more about that, there, you know, there were just some really cool answers to some really cool questions, and they just came from any direction. And one of the luxuries that I have in my position is we can really pursue just about anything we want. And as a result, we've been able to answer some cool questions that really drive at how form is, how function is derived from form and materials and behavior, as you've seen. In addition, you know, these students learn some really integrative techniques, modeling, instrumentation, data analysis. These are, I'll just reiterate, these are all exceptional people who led these projects, all undergraduates. I just want to stress that took a chance to learn some new skills that, you know, I'm not too sure many of these folks that I've worked with will ever swim fish or stretch skin again, but they learned a ton, some skills that they didn't expect to learn when they started their biology career at BC. So with that, I'd like to thank folks, especially Jen and Colin for having me here. And I'd like to point out Mike Sigler. I don't know if he's in the crowd or not, but I think Mike probably had a hand in getting me 2 shoals in this virtual format. Mike and I sailed on a cruise, I don't know, 15, 16, 17 years ago, and he's been a good friend ever since. I'd also like to thank all the other undergraduates in the lab who continue to do great work, and of course, Boston College for supporting it. So I think I could take questions. Yeah.
Yeah. Thank you so much, Chris. That was great. I just learned a lot. All right, so please ask your questions now. Please write it in the Q&A box, please, and I will read them out loud. And don't be shy. Chris has agreed if we don't get to all the questions, he'll answer them later. So you will get an answer, don't worry. So first up is Doug Fudge is asking, what kind of results from the skin tensile tests would have led you to conclude that the skin is not involved in force transmission?
Well, if I understand the question, if we had not found any discernible pattern in the stiffness across the body, in other words, if it was kind of a free-for-all for the stiffness, or if the skin was more stiff, say in the anterior. In other words, if we see this tendinous connection to the skin with the myotomes that becomes more important down the posterior of the body, at least in rayfin fishes and in those ones that we studied, and then we see the stiffness of the skin increase as well, that leads us to infer that it's a force transmission device. If we didn't see that stiffness, then we'd say, okay, well, the skin is just attached to these muscles via these tendons, but maybe they're not transmitting much force.
Oops, I think you just froze on us for a second.
Oh, did I? Am I back?
You're back, you're back.
Great.
I actually have a follow-up question to that, for that particular study, which I mean, this is totally my bird bias, okay? Just thinking about how a predator catches its prey isn't always necessarily the first thing it would eat. You know what I mean? Like, you inferred that the guts would be high value food part of the body of the fish.
Yeah.
It'd be really interesting to think about how the predators of those particular fish might attack them. Did you think about that at all?
We have, and we waded through some very old literature, some actually very clever ones, people putting out models and seeing where blue herons would strike at them. And that we really haven't followed up on it. Eva, who led this study, she just graduated and she just submitted her thesis, a few months ago. But that is a really great line of future research. Absolutely.
Well, Mike is here. He says hi, and he says, great talk. He's actually asking a question that I was thinking about, because I was thinking, if that dragonfish, the teeth are its cage, yep, and it's caging something up to around 50% of its body length, how the heck does it, I mean, obviously then those teeth have to have the strength, and the whole animal has to have the strength to hold that. That's incredible.
Here's that is another great question. There's so many unanswered things here, and that's it's sort of a problem with the deep sea. You can only do so much. It would be lovely to put a dragonfish and a myctophid in a tank and just see what happens, like we do for feeding studies in other more tractable groups. But it's a wonderful question. What I think might be happening here, and I have some thoughts on how to approach it. They're all very immature. The jaw probably doesn't close against a myctophid, and these teeth are really, really long in most of these dragonfishes. So the effective gape is, the jaw may open like this, but the teeth form an effective gape where it's actually not that big of a gate, so it might be able to cage around the animal pretty quickly. But then that doesn't answer your question about how does it hold on, right? And these are big fish; they swim pretty quickly, pretty powerfully. How they don't get through there, I will say we do have some data where the max prey size, the width of the body, in many of these dragonfishes we looked at, the width of the body is the width of those or the distance between those two interior teeth. So it might just be snug enough fit that they don't swim away. But that's just total armchair stuff.
All right, here's Noah is chiming in now. Noah Bryceman, wonderful talk, Chris. When doing your experiment with fish skin, did you ever notice a species with skin in high tension? I ask this because while filleting dissecting a pacu, I think I'm saying that right, the skin released in an extraordinary amount of tension upon the first cut compared to most fish that I've seen. And I'm curious if you've ever noticed this among your species.
Yes, matter of fact, that pompano. So that most of these, well, all the not just actinopterygians, just about every aquatic vertebrate, we're talking snakes, we're talking whales, have this helical pattern to the collagen fibers, cross helical, and it's sort of arranged like plywood, but uniformly. All right, and that that creates sort of this helical tension structure. And if that skin is really thick, and it was in the pompano, and I've dissected some whale skin before, it was the same thing. We cut a section out, and the skin just went, it shrunk right up. So yeah, and in some species, it is a pretty tense system. Yeah, and those tend to be those species with the stiffest skin.
Here's another question, referring to the swimming study, the last one. Did they have a chance to look at the relationship between wavelength and departure from midline or the amplitude? How they change toward the tail? That would seem to contribute to the thrust vector component.
Absolutely, and that is an important part of understanding this that I didn't focus on. So yes, the amplitude for this wave also to increase along the body, the amplitude has to increase as well. So if we look at this, all these red lines here in this figure, the middle figure here, all these red lines represent the amplitude of the midline waveform along the body. And what you see here is an envelope of that amplitude, just how high it is along the body. And you can just see it increases quite a bit down the body, right? So the amplitude is increasing in addition to the wavelength increasing. And that wavelength will increase if the amplitude is increasing and there's more shortening than there is lengthening in the wave. That's kind of the point there.
That's great. This last comment is kind of a request for you to come speak to an 8th grade class. So there's a teacher in one of our courses out here, and Donna Collins is saying how much she likes this research, and she does biomimicry projects with a 7th grade life science class. And this year, many projects include doing research on fish scales and skin and how the mechanics of each could be re-engineered into things to solve human problems. They would love to see this when they return for 8th grade in the fall. So maybe Donna can reach out to you over email.
Yes, please do, Donna. I would love to do that. That's very flattering. Very, very flattering. Yes, thank you.
Yeah. That's awesome. All right, well, that sums up our questions. Thank you so much, Chris, for that. So everyone, we have recorded this talk, and it will be on the Shoals live stream page on our website, so you can share with your friends or family, or go back and remember what you've learned here. Our next Rock Talk is next Thursday, the 8th, where Dr. Andrew Davis, assistant professor from Clarkson College in New York, will be with us. He works around the world to explore topics of aquatic species invasions and spread. He examines the influence of introduction events and climate change, and employs a diversity of research methods, just like Chris just did, that includes genetics and high-resolution particle tracking methods to look at species invasions. So that will be next Thursday. For more information about all of these talks this summer, you can go on our website and look under the seminar series. Thank you all so much. Thank you, Chris, for being with us tonight, and everyone have a great night.
Thank you.
Bye.